So students will talk about the Human Genome Project and DNA fingerprinting in this video lecture. So Human Genome Project was an international scientific program with the goal of determining the complete genome information of the human. And it was planned in 1984 and it was initiated in 1990 and it was declared completed on April 2003. It this project was funded by US government and National Institute of Health, in short we can say NIH, as well as numerous groups around the world. And it was a joint research program of different countries such as USA, United States of America, United Kingdom, Japan, France, Germany and China. And the whole project was completed in 13 years. So we will see what was the goal of Human Genome Project. So first uh, point is to identify the complete human DNA sequence, and it is around 3 billion. Second point is to identify the genes, and now we know that it is approximately 20,000 to 25,000 in human. And the third point of the Human Genome Project's goal was to create a genome sequence database to store the complete information for further use. Fourth point was to improve the tools for data analysis. Fifth was transfer related technology to other sectors, for example, industries, for commercial purposes. And the sixth point was taking care of legal, ethical, and social issues that the project may pose. So it was a project to identify and analyze the whole genome of the human. So we will see what was the methodologies the scientists took to complete this project. So two major approaches were followed at that time. First was ESTs, Express Sequence Tags. So ESTs were to identify all the genes that express as RNA reference as ESTs. It means EST means as we know that only 2% of the whole genome sequences transcribed, or we can say involved in the formation of protein. So uh, the first the first approach was to identify those genes to which expresses. So the gene which expresses is called as EST, Express Sequence Tag. So it is a method of cloning and sequencing of a set of only DNA sequence which is expressed. So what they did, they took the only those sequences which were expressing. The second method was annotation. So it was the process of making specific features in DNA, RNA, and protein sequence, which descriptive information about structure and function. So what they did first, they in case of ESTs, they took only those sequences which were expressing, and in case of annotation, they defined the that particular EST in terms of their structure and function. That was called as annotation. So we will see the salient features of Human Genome Project. So what they found was the human genome is made up of 3.1 into 10 to the power 9 bases. The size of gene ranges from 100 to millions of the bases. It means some of the genes are composed of hundreds of bases and some are composed of millions of bases. And the total number of genes was estimated as 30,000. And previously it was hypothesized that it might be 80,000 to 1 lakh 40,000 genes. But after estimation, they found that only 30,000, they found that only 30,000 genes are there. But it was the approximate amount. Some of the you can get information that somewhere it has been written 20 to 25,000. It means around 30,000 genes were there. So almost like the last point is the almost 99.9% of nucleotide bases are exactly same in all the people. It means there is very less difference among persons. And still, more than 40% of the discovered genome functions is still unknown, and less than 2% of the genome encodes for the protein. Chromosome 1 has the most of the gene, and chromosome Y has very less. Since chromosome Y is very smaller in structure, that's why they have a very less number of genes. They identified 1.4 million locations where SNPs, single nucleotide polymorphism, occurred. Now we will talk about DNA fingerprinting. So this technique has been used to compare the DNA sequences of two individuals, and each of a person has a unique genomic composition based on arrangement of nucleotides. It means each and every person is having a unique genomic uh, genomic basis composition. So our genome is composed of coding and non-coding sequences, and non-coding sequences are composed of regulatory sequences, intervening sequences, or we can say introns, and repetitive DNA sequences. So for the DNA fingerprinting, people used to focus on these non-coding sequences, and particularly the short repetitive sequences. So we can see here that genome is composed of two kinds of sequences. First is coding sequences which transcribe into protein, and the second one is non-coding sequences. It means it is not directly involved in the encoding of protein. So it is again divided into three categories. First one is regulatory sequences, means it regulates the expression of genes, and the second one is intron, which is present in uh, nascent mRNA. And the third and very important sequence is VNTR, variable number of tandem repeats, or repetitive sequences. And these repetitive sequences can further be categorized into two categories. First one is long repetitive sequences, and another one is short repetitive sequences. And these short repetitive sequences are very important for the DNA fingerprinting. So repetitive sequences are also called as VNTR, or variable number of tandem repeats, since it is a group of nucleotides in a particular arrangement are present in a repetitive fashion, and it is short set of sequences of DNA repeats are very important in determining DNA fingerprints of individuals. And the father of DNA fingerprinting is Alex Jeffrey, and father of DNA fingerprinting in India is Lalji Singh. So the DNA fingerprinting involves the identification of differences in these repetitive DNA sequences. It means uh, uh, DNA fingerprinting is all about to identify the differences of these repetitive sequences. So VNTR are three types: satellite, mini satellite, and microsatellite. It means the short repetitive sequences are again categorized into three categories: satellite, mini satellite, and microsatellite, based on the length of their repetitive sequences. And these repetitive sequences are very polymorphic and heritable DNA sequences. So the polymorphism in the DNA. So one question comes in our mind: what is the meaning of polymorphism? So polymorphism is the variation in DNA sequences among the individuals. So now we will see what is the what are the kinds of polymorphisms exist in human beings. So first polymorphism we can see here, SNP, single nucleotide polymorphism. So we can see here that there are two DNA sequences of different individuals, and there is uh, there is a change in nucleotide at a single position. We can see here cytosine is present in one individual and thymine is present in another individual. So it, this kind of polymorphism or variation in the DNA sequence is called a single nucleotide polymorphism. And the second is multiple nucleotide polymorphism. So we can see here that uh, there are two DNA sequences of two different individuals. So one is having this G80 and another one is uh, this sequence is absent. So this kind of multiple nucleotide polymorphism occurs due to deletion of this particular uh, codon. And the second type of multiple nucleotide polymorphism we can see here, the difference in the nucleotide sequences. We can see here that one person is having cytosine, 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 C, 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 and another individual's DNA sequence at the same site is AGTC. So we can see here that there is a difference in DNA among two individuals. So this is called as block substitution. Means the new or another kind of nucleotide substitutes a particular location, and another kind of variation is or multiple nucleotide polymorphism is called as inversion variant. So we can see here that it is inverted sequence. And the third point is polymorphism VNTR or variable number of tandem repeats. So we can see here that there is a GGCCTTA, one short sequence, and it has been repeated two times. We can see here GGCCTTA. We can see in one individual two repeats are there, but in another individual only one repeat is there. So this kind of polymorphism we can utilize for the DNA fingerprinting. So, so we can see here that in one individual, it is a repeat of these sequences two times, and another person, it is one times repeat. So these sequences have been utilized for the DNA fingerprinting and identification of two different individuals. So the process of DNA fingerprinting, we will see how how the scientists proceed the experiment to identify the different individuals based on DNA fingerprinting. So the first step is DNA extraction from the sample cells. It means first the first step of the DNA fingerprinting to extract the DNA from the individuals. And the second one is amplification of many copies of DNA sequence. It means what they what they are doing, they are using polymerase chain reaction to amplify the DNA sequences of individuals from which we have extracted the DNA. And the third point is or step is restriction digestion by utilizing uh, a restriction enzyme. And what is a restriction enzyme? Restriction enzyme is a type of special, we can say, molecular scissor which cuts the DNA at specific site. So what we are doing, we just cut the DNA at a specific point of the samples of the amplified DNA. The fourth point is separation of DNA sequences on gel electrophoresis. So here in case of gel electrophoresis, we used to run the cut DNA. And the fifth point is southern blot. And the name southern blot have been uh, kept as the discoverer of this southern blot. The name of the scientist was Southern. So here in southern blot, the separated DNA in gel electrophoresis is transferred exactly on the nylon membrane. And the sixth point is hybridization of these VNTRs present on the nylon membrane, as we have transferred all the DNA on the nylon membrane. So what we used to do, we utilize radio labeled probe, which is complementary to the particular VNTR, and rest of the non-hybrid or non-binded or non-hybridized DNA is washed off. And the next step is exposing the hybridized DNA of individual in autoradiography system on X-ray. So the last step is analysis of pattern of hybridization between individuals, and the exception is identical and monozygotic case. It means this DNA fingerprinting cannot be applicable for identical or monozygotic case because the genomic DNA structure is same in both the individuals, and its application is forensics. It means the this technique has been utilized in forensics, genetic relationship of an individual with their parents, and population and genetic diversity studies. So this schematic representation can be uh, can be used to understand completely what is the DNA fingerprinting and how people use to identify different DNA sequences. So we can see here as an example, three different chromosomes have been taken and three different repetitive sequences have also been taken. So we can see here that there are three repeats, there are four repeats, and in case of another individual, there are several repeats of these sequences. And for the another kind of repeat, we can see clearly that in among the allele and among the individuals, again there is the variation, and the same is also present in chromosome number 16. And after gel electrophoresis, we can see the different pattern of the DNA also. So we can see here, suppose this is the sample sequence or query sequence. So we have run this DNA in this gel. So this pattern came, and we took to identify the sample matching. So we took two different DNA sequences and we used gel electrophoresis. So we can see here the DNA pattern based on their size. So the smaller will be on the lower side, lower side of this gel, and the smaller will come on the upper side. So the larger sequence here, among all the sequences of two individuals, we can see here this red repetitive sequences has been representing here. So, and the smaller one is here. So on the basis of size of the fragments which were cut by using molecular scissor or restriction enzyme, we can see here that DNA is representing different patterns here. So on the basis of pattern, we can compare. We can say that C is exactly matching with B. So this kind of analysis we use to take to identify the DNA sequences during DNA fingerprinting. Thank you.